Okay, there we go. All right. So today we're going to start talking about some animal body plans as we move into the Cambrian, because the Cambrian is all about rapidly diversifying animal groups, basically. So we saw when we were talking about the Ediacaran, we had some soft-bodied fossils, but we weren't totally sure what they were doing. And we talked a little bit about their extinction. One of the big theories about why they went extinct may be the rise of the metazoans, which are the animals. So we're going to walk through some animal groups today. But before we do that, I want to make sure that as we step through a tree, let's make sure that we got the base of the tree covered. Okay, so when we are classifying our groups of organisms, we start with domains. So the current idea is that we have 3 domains of life. So we have bacteria. So those are our unicellular prokaryotes. We have them basically everywhere. We have another really different group of unicellular prokaryotes that we don't think about as often because they often live in really extreme environments. So for example, these hot springs, these colors are actually made by some organisms in this domain, by some archaeans. And finally, we have our domain eukarya, and we talked about the evolution of eukaryotes. So eukaryotes can be either unicellular or multicellular, but one of the main differences is that we're going to have membrane-bound organelles. So we have these internal compartments with specialized function. So specifically, our mitochondria for producing lots of ATP and chloroplasts for photosynthesis. Okay. So when we're comparing these domains of life, one of the ways that we tell them apart is by looking at some of their characteristics. So there's been debate about how to divide our groups of organisms when we're thinking about their phylogeny, their evolution. So one of the pieces of data we have is what we can observe about these groups. More recently, we've been using molecular techniques in order to investigate this further, and we'll see a tree based on rRNA sequences in a little bit. But some of the things that we could observe under a microscope or with some basic tests are in this table. So this gives us a great chance to practice some of our terms. So I want you to look at this table. I want you to find an apomorphy of the domain bacteria and one of the domain eukarya. I want you to look for a synapomorphy of archaea and eukarya. So who wants to remind me what an apomorphy is? Yeah, so a trait that's unique to a group or species. So who's got an apomorphy for domain bacteria? Peptidoglycan.
Yeah.
Yeah, all right. So in our bacteria, when we're looking at the cell wall, we can see peptidoglycan present in bacteria, but we don't see that in either the archaea or the eukarya. So that would be an apomorphy of that domain because it's a unique trait that we only see there. So we expect that it evolved due to some mutation that was present only in the last common ancestor of this group, but not in the other group. Okay. So what about domain eukarya? What apomorphy or apomorphies can we see there? Okay, yeah, so we see absence of a circular chromosome in eukarya. What else do we see in eukarya?
Envelope.
There can be a nuclear envelope. What else? Yeah, those membrane enclosed organelles. Okay. So these are all traits that we see only in the eukarya. So we're considering them apomorphies. A detail that we would want to put on there, when we think about an apomorphy, we usually think of it as a derived trait. So that's kind of embedded in the term. So when we evaluate these characteristics, to say for sure that it is an apomorphy, we would want to know that this is something that evolved within eukarya rather than something that didn't evolve. And I'm saying just looking at the circular chromosome, totally possible that losing a circular chromosome is an apomorphy in eukarya, but it could also be the case that we have some type of convergent evolution in bacteria and archaea, or that they share a more recent common ancestor. So apomorphies are about derived traits. With this data, we would think that all three of these are apomorphies. If we got additional data, we might revise our assessment. Okay. So let's take a look then at the synapomorphy. So remind me what synapomorphy is. If an apomorphy is a derived trait, a synapomorphy is going to be
The derived character
Yeah. So we also, when we're thinking about synapomorphies, we're going to be thinking about a group and their shared common ancestor having that trait as well. So if we think about a synapomorphy, if I'm telling you we have a synapomorphy for archaea and eukarya, this is implying a specific phylogeny. I'm implying that they have a shared common ancestor of those two groups specifically. So if that's true, where can we find a synapomorphy on this chart? So we have several kinds of RNA polymerase here. Do you see anything else?
Yeah.
Yep. So just the opposite of that bacteria apomorphy. Anything else? This thing? Yeah. So we also see that the initiator amino acid when we're making proteins in archaea and eukarya is the same. It's methionine. We got formal methionine.
So if you have something like this one where it's present in some species, so is it only some species that considered like important?
That's a great question. So what this implies is synapomorphy might not be the best term to use here. What this implies is that if we're going with this phylogenetic assumption that eukarya and archaea share a common ancestor, which we'll look at, I think it's on the next slide, this would imply that they evolved that histone trait, and then some of them lost it, essentially. So yes, maybe, but basically, yeah. So that might, yeah. That's what I have to say about that. Okay. So we have these three domains. They're pretty different. And we can make a couple of hypotheses or different ideas about how these three groups branched from each other. Now, one of these is currently more supported than the others. We do currently perform one of these hypotheses, but we can use this also to practice our terminology. So go ahead and go through each of these and think about what we have as sister groups and what we can think about the last common ancestor for each of these groups. I'm going to give you a second to look at these trees and think about how they're different and how they're potentially the same for some cases. For the sister groups question, what could we say about sister groups in general? Do we have similar sister groups in any of these 4 hypotheses? Similar assumptions or similar findings, I guess. What do we see here? Yeah. So 2 of our hypotheses, we see 2, 4. Both have archaea and eukaryote as sister groups. Hypothesis 1 also kind of does. But in hypothesis 1, we also see bacteria as a 3rd sister group here. Right, so we got 1, 2, 3 sisters. And this tree doesn't have an assumption at all really about where this tree is rooted. So when we look at trees and they have this little stem at the bottom, we call this a rooted tree. So this is unrooted. It's just kind of branching from a central point. Since we are looking at that one, okay, what do you think this would imply about the last common ancestor when we're looking at this hypothesis 1? What could you say about the last common ancestor of the group? If I were going to draw on a last common ancestor, where would I put it for hypothesis 1? Yeah, I just put it in the center, right? So in this hypothesis, we're seeing them all branching separately from this last common ancestor. So the implication here is that we had a last common ancestor that was none of these 3 things exactly, but they all separated out from something that was kind of in between, something that was older and different enough from all of these so that it's not fair to group it with any one of them. So where can we see a hypothesis that makes a different statement about the last common ancestor? How about hypothesis 4? What do we see in hypothesis 4? If you had to think about the last common ancestor of the 3 groups, what would you say about it if you're looking at hypothesis 4? So the last common ancestor of archaea and eukaryote would be here, right? So yeah, that would say this last common ancestor of archaea and eukaryote essentially was a bacterium, right? You can see bacteria is just like on that line. So this is saying that bacteria were first, and then a bacterium split into these two groups. So we have mutations developing, changing, and then these two groups branching off. So this would also imply that really we kind of start with bacteria here. So what do we see that's different about hypothesis 3? Yeah. So what we see here is the assumption that we had some organism that was something like bacteria or archaea, not quite either, right? Which makes sense. We're both unicellular prokaryotes, right? So we think we started with some unicellular prokaryote, right? But then we had a split forming bacteria and our archaea, which are these sort of streamophiles with some differences specifically in their membranes and some of their internal cellular processes. And then the eukaryota evolved from something that came back to. So when we say a phylogeny is a hypothesis sometimes, or we can treat phylogenies, different phylogenies as hypotheses, this is kind of why, right? So these are each just a picture, but each of them has nested within them a lot of testable assumptions. So you can almost think of them as like a big collection of hypotheses, right? Because they make statements about our last common ancestor, they make statements about how these groups are related to each other, make statements about when these branches happen, sometimes depending on the phylogeny. You could also note that in hypothesis 3, this would be a different example of sister groups, right? So here we can see bacteria and archaea are actually sisters. The most commonly cited of these hypotheses, so the current most popular one, best evidence for our arrangement is this hypothesis 2. So our current understanding, our best guess, is that we had a last common ancestor, something like bacteria, something like a mix of the archaea and the eukaryote. So we had something kind of different than all three of them, kind of like this hypothesis 1. But then bacteria branched off separately from the ancestor to archaea and eukaryota. And then they separately have a common ancestor to just those two that branched into those separate traits. So we would expect that evolution of multicellularity to happen somewhere here. So we know that the difference about the eukaryotes, we look back at that table, right? Two of the big things up at the top of the list for eukaryotes were a membrane-bound nucleus and those membrane-bound organelles. So when we talked about the eukaryotes, right, we were saying that we have some unicellular prokaryote of some type, and then some smaller, give it a different color, some smaller, different unicellular prokaryote. And the idea is that one engulfs the other, right? So we slowly take it in, maybe we're trying to eat it, who knows exactly why. Instead of digesting, we end up with this internal membrane, right? So this is the idea for how we end up with those membrane-bound organelles, because each of these had their own membrane to begin with, right? So here we see a cell membrane, here we see a cell membrane, and we maintain those cell membranes in our eukaryotic cell. That's why we end up with those individual membranes. So within this hypothesis 2, we would expect that this happened somewhere after the branching between the archaea and the eukaryota, because we don't see those organelles in archaea. we do see them in eukaryotic. So somewhere along, which then also gives rise to our ability to be multicellular, right? Our eukaryotes are the only multicellular group, so we would expect that to be somewhere along here as well. So questions about these hypotheses, how to look at them, how to think about what they mean for the relationships between those groups. So here we see a phylogenetic tree based on sequencing of rRNA. So that's ribosomal RNA, so our ribosomes are proteins. So we have ribosomes in all of our domains, and we can sequence them, look at that rRNA, see where, in which groups are they more similar, in which groups are they less similar. And when we do that, this is the tree that they ended up with, which is part of how we came up with this idea that we have 3 domains in the first place. All right, so we can see the bacteria here separating out first. Then we can see that the archaea and the eukarya have slightly more similar rRNA among all of those groups. And we can see that there's a split kind of down here at the base. So that's why we like that hypothesis 2 best currently. It's based on basically this data where that comes from. Okay. However, this doesn't necessarily map on amazingly well to the type of descriptions of kingdoms that you'll see in biology textbooks. It gets more and more complicated essentially as we get more and more data, especially this molecular data. So in your traditional biology book, you'll see some groups of kingdoms, and sometimes it'll be as few as 3, sometimes it's as many as 8. I think the most common is these 6 traditionally recognized kingdoms. So I want you to take a look at this phylogenetic tree of rRNA and figure out what the relationships of these groups would be if we just pick those out. We want to draw our own phylogenetic tree for those kingdoms. Okay. I gave you an example of a protist, like an amoeba. Because there's nothing on the thing. This phylogeny that obviously says protist. So take a second to look at this and try and draw yourself a tree of how you would relate these on this chart, I'm going to go ahead and start circling, and then we can build our tree together. Bacteria, new bacteria. For Archaebacteria, we're going to pick all our Archaea. Excellent. So we can start by creating that split. We start with a common ancestor. We have our bacteria branching off. Bacteria. All right. Then we have our next branch, which is leading us to this split point of our archaea and our eukarya. We'll put our archaea here. Okay. So let's keep moving on up. What are we going to put at this next branching point? What's the next group that's going to go off by itself? Yeah, at least with the example I gave you. That's, and now we're going to have our Protus. Protists are actually weird and maybe not a monophyletic group for real, but I'm going to leave them there. Okay. So what do we have happening next? So we're going to have our plants split off. Phylogeny is not beautiful because I'm doing it in the corner. This is presumably better. Okay. And so then our final sister groups here at the top. Yeah, got our fungi. Is anything about this tree surprising to you? Specifically, because I also teach plant systematics, and even though I say it a lot of times, I inevitably get a mushroom as a plant example in people's collections. So my impression is that not quite everybody realizes this last split, right? Fungi, mushrooms look kind of like plants, they like don't move when we eat them, right? But they are actually closer to animals when you look at evolutionary needs. I've branched off separately. So this is the general relationship. If I were drawing a tree super properly, when we have species that are all alive at the same time, we do like their branches to all come to the same level. So like I drew it like this so that we'd have space to write, but technically like all the tips should come up all the way to the end, since all of these groups are still present in the modern day. So as we move into the Cambrian, we think a lot about the animals because we start to see all the major body parts that we see in animals suddenly appearing in the fossil record, basically. Okay, so I want to place us in time here. So here we're looking at a phylogeny that also gives us when we start to see all of these groups. Okay, so we're thinking about the Cambrian. Whereabouts would you kind of put this? And also, since this is on our chronostratigraphic chart, does everybody know how to convert this to this GA timescale from millions of years to the billions of years? So this is just a note that that is 0.5 billion years. So that would be 0.5 GA. So we're going to be somewhere, let's say, like in this time period. We don't have that time exactly on the scale, but we're going to be thinking a lot about our early animals, which we saw that we have some of the very early groups of metazoan, starting in the Ediacaran, so right before the Cambrian. We saw that that was maybe why we see the disappearance of the Ediacaran biota that we were talking about at the end of our last unit. So we have a little bit of this starting there, but this is where we really start to see the diversification. So since this tree also has time in it, making sure we can see which group here is older, the plants or the animals. Yeah, here we have the animal as older, right? So we could still think in terms of that previous tree, right? Like we might want to put fungi over here, right? But it's drawn like this so that we can put this time scale on it basically. Right? But what a thing we want to realize is that we are not actually to plants yet. So we have some photosynthesis going on because we have things like cyanobacteria, but we don't actually have any true plants when we're in the Cambrian. So that's why we're talking about animals and not plants. Not just because animals are fun to talk about, but plants kind of don't exist yet. So during the Cambrian period, we suddenly see tons and tons of fossilized organisms. So there are debates about like why we suddenly see this in the Cambrian period. When they first found the rocks of the Cambrian period, they kind of thought that this was like the beginning of Earth, the beginning of life. So all of it appearing all at once. We now have data that shows us that life was probably present before this time period, right? So we found some new fossils, we found those Ediacarans, but we've also now had the ability to slice up rock really tiny and look at microfossils so we can see that evidence of early unicellular life as well. But during the Cambrian, we now have a lot of physical fossils because we're now going to have basically hard body parts as well. So this is an improvement for our fossilization process because we have some structures that are a little bit more likely to stick around. Most things like don't stick around in the fossil record because most organisms, when they die, decay and don't end up fossilized. But because of some of these new traits that we see in our evolving metazoans, we have a little better fossilization. So we see some major groups appearing. So we can see arthropods appearing, members of really all the other modern groups of animals, as well as some weird stuff that is no longer alive. Right? So some stuff with some weird experiments that did not survive through to the present day. And here we see an example of what some of these fossils look like, and we'll look at some of them in greater detail in future lectures, so probably on Wednesday. All these arrows are actually pointing out eyes. So as we think about the Cambrian, we'll also think about the development of eyes in a little bit. Now, another evolutionary concept that this is a great place to think about is when we think about evolutionary speed and how evolution happens. So before, when we talked about evolution, we mentioned we're talking about mutations, we're talking about speciation. So what we're talking about is mutations collecting over time until groups become so different from each other that they are considered new species, and so different from each other that they can't interbreed, basically, is one species concept. And we can talk more about species concept as we move forward. There'll be some iffy points as we go through the history of life. There's actually been a huge argument in the past between people who think that evolution happens gradually, right? So we have a really slow accumulation of mutations, right? The slow progression until eventually we're different. So you can see here at the split, we have very similar organisms. And then we have this continued sequence, this anagenesis, as we continue down the line. So in gradualism theory is that evolution occurs with slow accumulation of small changes. This is really what Darwin proposed in The Origin of Species. However, when we look at strata, so layers of rock, like those from the Cambrian, we see that we suddenly have all these different forms. So one of the big arguments is with proponents of this theory of punctuated equilibrium. So punctuated like periods, like kind of stop and go. So people who prefer this punctuated equilibrium model think that what's happening is that we have long periods where everything kind of stays the same, but we have short periods where something changes and we end up with really rapid evolution from a geologic time scale, right? Like not in terms of our lives rapid, but geology rapid. So in punctuated equilibrium, we expect our splits to kind of be close together and to sort of rapidly see this appearance of new organisms, but then kind of continue in similar form for a much longer period. Now, the reality is, right, we don't necessarily have to choose just one of these models, right? We can expect that in certain situations, we have more gradual change. In certain situations, maybe we have some of this punctuated equilibrium, and really maybe we have some combination of both, right? Like this gradualism happening and occasional. But these are common terms that you'll see when you see people talk about evolution, this idea of gradualism versus punctuated equilibrium. And that really dates back in history to when people were looking at this sudden appearance of all these Cambrian creatures, trying to figure out how that could possibly be explained by gradualism when we don't see something ancestral to this slowly changing over time. We suddenly see all these early animals essentially appear. So when I say that we have all sorts of body plans appearing, basically animal phyla are separated out based on their physical shape, essentially. Now, some of this has to do with their physical shape in development, but you can find key features of their body plans. And sometimes in old literature or pretentious literature, you'll see this written in German, Bauplan. Okay. So these major phyla are grouped out based on physically how their shape is. And this does make sense a little bit from more of a developmental perspective than it first seems, right? At first, it seems like maybe somebody was just sitting there and sorting them based on what looks the most similar. which we do, right? Like, that's part of it. But if you think about body plan, it's really hard, right, to change something about fundamentally the way the body is structured with a mutation and not end up with an organism just dying off, right? So like, if you suddenly don't actually develop a spine and bilateral symmetry as you are developing, that is not going to go well for you. So when we think about body plans and different body plans, this is somewhere where we would expect mutations kind of like not to accumulate very often, just because of the crazy detrimental effects mutations to development of the shape of the body would have. Like if you suddenly start trying to develop like a mollusk, like a snail or something, that's not going to be a beneficial mutation for you. Okay. All right. So we're looking at these body plans, which is part of what we were looking at with this chart on the end, at the end of Friday. So we're looking at the Cambrian explosion, and we're seeing how all these different groups are arising here in the Cambrian. All right, so we saw this mapped onto some of our glaciation events. We talked about the snowball Earth theory. I think mainly refers to the glaciation in the Cryogenian, but we do have other glaciation events. So multiple times that we've had the Earth kind of covered in ice. Okay. So what we were looking at here was we saw a common ancestor to our metazoans. So metazoans, it's a fancy word for animals, right? A fancy science word for animals, metazoans. Okay. So we then see progressive branching as we go from the Cryogenian into the Ediacaran and then finally into the Cambrian. So exactly where these splits happen is kind of subject to debate, but given that we see examples of all these major groups in the Cambrian, and we don't really see intermediate fossils, right? Like we don't see transitional fossils somewhere in between all of these during the Cambrian. We can't track that gradualism. They just kind of suddenly all appear. What this really means, or what we theorized, right, is that those splits were happening sometime in the Ediacaran early on, and either they weren't happening in places that have a good fossil record, right? So the geology wasn't right, or maybe this starts happening with soft-bodied organisms, like decay super rapidly. So again, we don't have great fossilization of those, or that there were just so few of them compared to the Ediacaran biota that were prevalent at the time, that just because there weren't enough of them, we don't see them in our very limited fossil record. And some of our genetic data does also suggest that these splits did happen quite a long time ago, earlier though. So we can start to see those splits towards these modern groups. The metazoa we saw here is a description word for all of our animals, essentially. And so we can see our periphera, our sponges just kind of existing on their own here, doing their own thing. So they're weirdos, but the rest of our groups, we can see we have a theoretical last common ancestor sometime in the Cryogenian. Then we have progressive splitting in the Ediacaran, and then we start to see them actually fossilizing and becoming prevalent in the Cambrian. So when we're thinking about metazoans, we do want to clarify a little bit about what makes something an animal. So metazoans, animals, are within our domain Eukaryota, so they're eukaryotes. They don't have a cell wall. We're going to have multicellular animals. Actually, a weird thing that happens to be true about animals is that they all have like collagen present in them. So if you break them down on that sort of molecular level. We got collagen in animals. Animals are heterotrophic. So hetero meaning different. Trophic, forget what the root means, but basically it refers to like eating and where they get their energy. So animals have to get energy by eating other things because they can't photosynthesize, they don't make their own energy from sunlight. So they're going to have to eat other stuff. So heterotrophic. Other things that tend to be true about animals is they're usually motile, so they can move, right? They're not the only thing that can move, but they're a best example of things that can move. Other things are usually weird exceptions when we're thinking about multicellular organisms anyway. And we see special sensory organs of a variety of types in our animals. So we're gonna step in then to think about our Bilateria. Okay. So this group, the Bilateria, is referring to the bulk of our metazoans. So most of them are bilaterian, okay, which refers to the fact that they have bilateral symmetry. So that's where that word comes from for this monophyletic group. Okay, so monophyletic because they're all monophyletic on this part. Yeah, right, they are. Okay. Cool. So monophyletic, we have this last common ancestor, and it includes all the descendants of that last common ancestor and nothing else. Okay. So when we say bilateral symmetry, what we're saying is if you put a mirror down the center of the body, you'd end up with identical right and left side. So we got a mirror image across the central axis. Okay. And this can be separated from some other organisms that have radial symmetry. So coral is an animal, but it doesn't have bilateral symmetry. That it's got this kind of star-like symmetry. And sponges just like are not symmetrical, period, even though they still go along with our metazoa. So as we develop this mirror image body plan with bilateral symmetry, this means that we now have a line with a front end and a back end, basically, right? So we have structures that are going to run down this midline. So we refer to the central line and its endpoints, usually as a rostral caudal axis. So rostral or rostrum refers to the face. So rostral is like a head end. Caudal refers to a tail. Caudal end, but here. Okay. Caudal end. We're going to have the central axis, which means that we have a front and a back end. And other terms to know about like how we're describing where stuff is on an animal. So we have two other terms, ventral and dorsal, that you'll commonly see. So ventral refers to the belly side of the organism, basically. So your ventral side would be the front of your body, so where your face and your belly are facing. The dorsal side, like the dorsal fin on a shark, the dorsal side is the back. So when we're thinking about bilaterans, we've got this central axis, and it has a front end and a back end. Right? So most of our bilaterans are going to have their sensory organs and their nerve centers centered at this rostral end. So basically what we're saying is we form a head at the head end, okay, rather than at the caudal end, the tail end. So it's a common basic body plan, but we can get more specific, unfortunately. So our next breakdown is within those two groups, or sorry, within the bilateria, is going to be between the deuterostomes and the protostomes. And all this has to do with is whether the anus or the mouth develops first. So during development, what happens is you have a collection of cells that's growing. Certain stage, it's called a blastula. And then we start to have a phase called gastrulation, where we have the development of the gut tube that's going to run down the center of the body. So you can see here the gut forming in both of these as this hollow part in the center, hollow part in the center. And we have different specialized cell types that form in layers kind of around this that do eventually give rise to specific parts of your body, specific types of tissue. So if you ever look into development in detail, you'll find out what type of cell all your nerves are derived from, things like that. Okay, but during gastrulation, one of these first phases, we're forming a gut. Okay. So the difference between the deuterostomes and the protostomes is just which end forms first. Okay. And in the deuterostomes, we see the anus forming first, and in the protostomes, we see them forming. Okay. So we're going to talk about deuterostomes first, because we are deuterostomes. So within our deuterostomes, you can see deuterostomes, deuterostomia, our sister to protostomia. You can see that split is happening before our Cambrian, but not a ton before our Cambrian. And we're going to start by talking about our chordates. So phylum Chordata. So when we're talking about chordates, of which we are an example, they're going to have 5 synapomorphies. Okay, so our common ancestor had them, and we see them in all our modern groups. So they're listed below, and they do have a fair bit to do with embryology and development as well. So we can see the development of something called a notochord. So we can see that in brown here. Up above it, on the dorsal side, we can see a hollow dorsal nerve cord forming. We have a tail that is positioned after the anus, so this post-anal tail. So we have additional tissue here forming past the end of the gut tube. We have pharyngeal slits. So these are basically like gills. You can see this is a very fish-shaped example. We have an endostyle, which is a feeding type apparatus. And for some more examples, because those words are pretty dry. So in humans, each of those becomes something slightly more familiar. So the notochord basically develops into your spine in humans and in vertebrates generally. So vertebrates are things that have vertebrae, so bony spine basically. Okay. That hollow dorsal nerve cord becomes the spinal cord itself. So like the nervous system running through the center of your spine, or from your brain out to the rest of your body. Those pharyngeal slits do start as gills, which are actually present in early stages of human development. The gill slits are actually important when you think about development of the head and neck. Each of the little sort of pharyngeal arches and pouches that exist during development become very specific parts of your face, your head and neck, and your brain. And each of these slits and pouches is actually associated with a specific nerve that goes to all of the structures that form from that slit. So we don't have gills in adult life, but in embryology, we do see gill slits. The post-anal tail, also what, I don't know. Right. The post-anal tail becomes additional musculature, and that's better as an example in fish. Oh, I think that was supposed to be also embryological as well covered over there. So we can see that tail here in these stages of human embryo development. So you can see early on we have a tail that we gradually lose. We do have a little bit of a coccyx, a little bit of a tailbone, but no real post-anal tail in adult life. Finally, the endostyle, not super obvious in us invertebrates, but in other chordates, that's filter feeding, In us, it actually became the thyroid gland. So that's what happens in a lot of vertebrates, tetrapods in general, or four-footed organisms. Since we just brought up a slide on embryology, there's another common phrase in evolution. When people talk about evolution, you'll often see this phrase, ontogeny recapitulates phylogeny. Ontogeny means development. So this idea is that the developmental progression during embryological development basically shows you the evolutionary history kind of that organism. Now, some people use this as like a hard theory. This is actually proposed as like a definite theory of how things work. I think it's better to think about this as more of an observation of things that is generally true than to use it as a rule that this has to happen. But you can think about why this might be the case when you think about body plans, right? And why mutations to our very structured body plan would be a problem, right? So if we have some mutation early in development that really changes the structure of that development, you see how that would more than likely be a problem as an embryo and a fetus and then an adult develop, right? So these early stages of life are really important because if they go wrong, it's much easier for everything to go very wrong. So we can see that when we compare the embryos of a bunch of different groups. So at the bottom here, we have humans. At the top, we have fish. We also got chickens, turtles, and salamanders. So you can see that the very early stages look very similar. So we can see those gill slits, for example, on all of them. You can see that post-anal tail. You can see a head end and a tail end. Right. Our fish and salamanders are more different from us, so they become more different sooner on. You can see that our chicken, turtle, and human are staying similar for longer until they eventually differentiate from each other. So you see that phrase? That's what they're talking about. And this is a cool thing to observe. So this is a way we can kind of get clues about evolutionary history of an organism. There were some other groups that we're looking at. This one's actually not that important, but in a couple of our groups, we're going to be thinking about how many segments we have. So in a group called Hemichordata, we have 3 sort of segments, so a tripartite body plan. I'm just going to leave that. In Echinodermata, in adults, we see radial symmetry, which is unfortunate because these are still bilaterian. Okay, so radial symmetry was when we have that kind of star-shaped symmetry. Okay. But even though we have that body plan, that radial body plan in adults, during development, we actually start out, even in the echinoderms, from these bilaterally shaped embryos, basically. So that's part of how we see that evolutionary history, even though we kind of converge upon this radial symmetry property in the adults. So starfish is an example of an echinoderm. We see radial symmetry. Another cool thing that we can see in our echinoderms is a water-based vascular system. So starfish take water in through here, through the structure labeled as the madreporite. And this is actually how they move. They basically pump water through these structures that run down each of their arms and even into their little tube feet. And based on the pressure of water that gets pumped into each limb, it changes shape, and that's what allows them to actually walk across the seafloor. Okay, we will pick up here next time.